Ferid Murad, MD, Ph.D., won the Nobel Prize in Physiology or Medicine in 1998 for his work with nitric oxide as a cell signaling molecule in the cardiovascular system. He and Robert F. Furchgott and Louis Ignarro, co-recipients of the award, discovered how this colorless gas is used to signal blood vessels to relax and widen them. Nitric oxide now, as almost everybody in the audience knows, is recognized to have broad applications ranging from treatment for heart disease and shock to reducing the possibility of pulmonary hypertension in premature babies. Dr. Murad currently is Regional Professor on the School of Medicine, Department of Integrative Biology and Pharmacology at the University of Texas Health Science Center in Houston. Dr. Murad's research with nitric oxide began more than 30 years ago when he was on the faculty at the University of Virginia School of Medicine. Despite initial disbelief by some of his contemporaries, Dr. Murad persevered in his conviction that this short-lived gas was crucially important to the body's signaling system. In the 1970s, Dr. Murad discovered that nitroglycerin relieves chest pain in heart patients by releasing nitric oxide. And by 1992, Science magazine had named NO its molecule of the year. Raises the question, what are the molecules of the year for the succeeding years? I'm not up to date on that. Maybe some of you are. Dr. Murad earned his undergraduate degree in pre-medical science and chemistry from DePaul University in 1958, and his MD and PhD in pharmacology right here at what was then called Western Reserve University in 1965. He completed an internship and residency in medicine at the Massachusetts General Hospital in Boston, and subsequently spent several years at the National Institute of Health. He was on the faculty at the University of Virginia School of Medicine from 1970 to 1981, reaching the rank of professor. Then he joined the Palo Alto Veterans Hospital, a Stanford University affiliate, in 1981, and later was appointed chair of medicine at Stanford. In 1988, he left academia to become vice president at Abbott Laboratories, in charge of pharmaceutical research and development. And in 1993, he left Abbott to found his own molecular corporation. In 1997, Dr. Murad became the University of Texas Houston's first chair of the Department of Integrative Biology, Pharmacology, and Physiology. As you might expect, he has been recognized and awarded many prestigious awards, including membership in the National Academy of Sciences, membership in the Institute of Medicine, and a fellow of the American Academy of Arts and Sciences. Also, he's received the Albert and Mary Lasker Basic Medical Research Award, the American Heart Association SIBA Award, the Baxter Award for Distinguished Research from the Association of American Medical Colleges, and as I've already indicated, the Nobel Prize. Finally, it's been my privilege to serve on the Board of Trustees for this university for several years with Fred Murad, and I have appreciated that opportunity. So please welcome to the podium Dr. Fred Murad.
Well, it's always a pleasure to return to Cleveland and see how this place keeps changing and changing in the University Circle and the Medical Center and the Cleveland Clinic. I think the Cleveland Clinic is almost like the cancer that MD Anderson is in the Texas Medical Center with all of its land acquisitions and buildings. But it's always fun to come back, and I do come back frequently, as you just heard. I developed my long-term research interests here as a student. I was fortunate in being one of the early students in the MD PhD program, and I worked in the laboratories of Earl Sutherland, chairman of pharmacology at that point, and Ted Rall, a young assistant professor and colleague of Earl's who helped look after his laboratory. They had just discovered cyclic AMP about a year before I came as a student. And it was quite an exciting time to see this new intracellular messenger molecule mediate the effects of so many drugs and so many hormones. It was quite exciting to see that happen. And that continued as an interest for all of these years. I've been interested in how cells communicate. We call it cell signaling, and how they do it through small intracellular second messenger molecules. And it's taken us into a variety of directions, some of it very, very fundamental. But we've always got in mind the notion of where this will lead us in terms of diseases and potential drug therapy. And it's become a gold mine for drug discovery, and I'll try to summarize this for you today. Our first publication with Cyclic, GMP, and nitric oxide was in 1977, when as you've heard, we discovered that some drugs, such as nitroglycerin, worked by forming nitric oxide to cause its biological effects on smooth muscle, blood vessels, etc. Today, if you look at PubMed, there are 81,000 publications in the field. There are very few disciplines in biology that could have more publications than that. Cholesterol, I think, is up to 140,000. Cyclic A and P, I think, is about 110, 100,000. G proteins is only 40 or 50,000. And I love to stab my colleague Al Gilman, because he too was a student here with me in those days. So it's been remarkable. There's no way in the world in the next 40 or 50 minutes that I can really summarize all the field for you, but I'll give you a flavor and hopefully get you excited to see the directions that it might be going. The concept of cell communication is really a rather old concept. Pavlov was a Russian physician, physiologist working in St. Petersburg, who had a patient with a gunshot wound to his abdomen. He noticed that when the patient would see or smell food, he would enhance his gastric secretions. This led Pavlov to develop the Pavlovian dog model, and he conditioned these dogs, if you remember from your psychology classes. He created pouches and fistulas in these dogs, would show them food, they'd enhance their gastric secretion, and at the same time he'd ring a bell. And after a period of time, the animals became conditioned such that he could ring the bell without showing them the food and also enhance gastric secretions. That resulted in Pavlov receiving the Nobel Prize in 1904 for understanding gastrointestinal physiology and how the senses and the brain talk to the stomach, a very major concept. Today we know that lots of cells talk to each other, and that's summarized for you in this first model cartoon. This represents 3 different populations of cells that are going to communicate with each other. And they could be any cell types, it doesn't matter. But let's call cell 1 a neuronal cell. Central neuron or peripheral neuron, doesn't matter. Let's call cell 2 an endothelial cell lining a blood vessel, and cell 3 a smooth muscle cell in the wall of that blood vessel. Cell 1 is going to talk to cell 2. It does it by making messengers. Earl Sutherland called them first messengers. Most of us today call them neurotransmitters. We call them hormones. We call them autocoids. We call them paracrine substances. We call them growth factors. We call them cytokines. They're given all sorts of names, and they come in all sorts of shapes and sizes and flavors. Some are simple amino acids. Some are very complex peptides or even proteins, such as the gonadotropins. These messengers are released into the bloodstream. Very often they associate with carrier proteins, specific binding proteins or albumin or others, to protect them from degradation. So they can seek out and home to their target site. How do they identify that target site? That target site has another protein embedded in the membrane often of that cell. It can be intracellular, but most often it's on the cell surface, a transmembrane protein. We call those proteins receptors. They are selective for certain first messengers or ligands. The conformational fit between the first messenger and its receptor is what declares the specificity of that interaction. If they don't have the right shape and counterform, they don't interact. A key won't open all the locks, it only opens a certain lock. Once this interaction takes place, there are intracellular molecules that often accumulate that Earl Sutherland called intracellular second messengers. And the first such molecule was cyclic AMP. They discovered cyclic AMP looking for the molecular mechanism of action of epinephrine, adrenaline, and glucagon to cause glycogenolysis to elevate blood glucose levels. And they found this heat-stable molecule that they purified, turned out to be cyclic AMP. Well, it turns out that the messenger or ligand that binds to its receptor doesn't necessarily have to go inside of the cell. All it has to do is tickle that receptor, and then a cascade of biochemistry ensues to result in either the enhanced production or accumulation of these intracellular messengers. Well, over the years now, other intracellular messengers have popped up. Cyclic GMP, calcium, eicosanoids, peptides. And while there are hundreds and hundreds and hundreds of first messengers, there are a modest number of intracellular second messengers, perhaps no more than 8 or 10 or maybe 12, and I'm sure there'll be some more in the future as well. Nitric oxide is a very unique intracellular second messenger. A simple molecule, a free radical with an unshared electron in its outer shell, and it's very lipid soluble, unlike other messengers, all of which are charged at physiologic pH. Therefore, to traverse lipid bilayers and other membranes, they need carrier proteins or channels or the expenditure of energy to make that transfer. That's not true with nitric oxide. Nitric oxide can go wherever it wants with no restrictions. It can travel up to a couple hundred microns before it collides with something and gets sucked up or scavenged by a thiol or a transition metal or something else. So it can function intracellularly in the cell in which it's made, but it also can be a messenger molecule itself to transmit information to neighboring cells, or I suspect also to distant cells. You can form complexes of NO with proteins, albumin, hemoglobin, glutathione, and maybe it's carried off to a distance and it dissociates again when it sees the right molecule that has a higher affinity for it than its carrier. So it's also a hormone. There's no molecule that fulfills all of these criteria. In the blood vessel, what it does is go next door to the smooth muscle cell and influence the production of yet another messenger, cyclic GMP, and we'll talk a lot about that. The soluble Guanylate Cyclase is the receptor for nitric oxide because it has a heme prosthetic group, which we'll talk more about. When that cyclic GMP is elevated in the neighboring smooth muscle cell, it activates a kinase, phosphorylates proteins, alters intracellular calcium, and alters the phosphorylation of contractile proteins such as myosin light chain. And that then is the mechanism of relaxation in smooth muscle. Now, nitric oxide, I believe, is a very, very old molecule. I think it participated in evolution. It's impossible to prove that. I wish I could, so I could go back to Stockholm again. But I think that as cells began to evolve and communicate and phagocytize each other and colonize with each other, they had to have messengers. And they were obviously very, very simple messengers. And I think nitric oxide is certainly a candidate, and perhaps other molecules such as calcium and phosphate, hydrogen sulfide, carbon oxide. So these were messengers probably before there was oxygen in the atmosphere 4 billion years ago. But it became important about 50 or 60 years ago when we recognized that most of the fossil fuels, or any substance that possesses nitrogen, when it's combusted in the presence of oxygen, makes a family of nitrogen oxides. And there are a variety of these. There's NO, there's N2O, there's NO2, N2O3, etc. These are called nitrogen oxides or NOxes, as you might see in the literature. They're pollutants that come out of automobile exhaust, cigarette smoke, smokestacks. And the environmentalists were all concerned about these pollutants because they resulted in the depletion of the ozone layer to cause global warming along with some of the greenhouse gases. But I'm going to tell you that they're also very important because they mediate the effects of some very, very important drugs such as nitroglycerin, nitroprusside, and other drugs as well. And they're natural substances in the body. This is a partial list of some of the biological effects of nitrogen oxide, the monooxide as a free radical. And I won't go through these because I'm going to talk more about them as we go along in the discussion. But they range from blood pressure, vasodilatation, to penile erection, to toxicity, to invading organisms, pathogens, cancer, cytotoxicity, et cetera, et cetera. Cyclic GMP was discovered in 1963, about 7 years after the discovery of cyclic AMP, by a couple of chemists who gave radioactive P32 to rats, collected the urine to measure and examine the major organic phosphates of the urine. They found two primary major organic phosphates, cyclic AMP and the second being cyclic GMP. That was the first demonstration that cyclic GMP was a natural product. Cyclic AMP research was moving along at a pretty rapid pace at this point. We all knew how important it was going to become here in Cleveland, and others began to become interested as well. But we didn't know anything about cyclic GMP. But this work stimulated several laboratories around the country to start looking for the enzymes that made cyclic GMP, enzymes that might inactivate cyclic GMP, and proteins that might be, quotes, receptors or targets for cyclic GMP in biology. We know today that there are a family of guanylyl cyclases that make cyclic GMP from GDP. All of the guanylyl cyclases, soluble and particulate, there are about 7 or 8 isoforms, have similarity in homology in their catalytic domains. They also have homology with the adenylyl cyclases because they basically catalyze the same reaction to convert a triphosphate to a cyclic nucleotide. In fact, if you do a mutation of one amino acid in the catalytic domain, you can convert one cyclase to another, an adenylyl cyclase to a guanylate cyclase or vice versa. The other product of the reaction is pyrophosphate. The cyclic nucleotide, whether it be cyclic AMP or cyclic GMP, has a high energy bond at the 3' ribosyl bond back on the sugar. The molecule is inactivated by a family of enzymes called cyclic nucleotide phosphodiesterases. It cleaves this phosphodiester bond to form the corresponding monophosphate, either 5' AMP or 5' GMP. There are at least 11 genes for phosphodiesterase and a variety of splice variants. It's going to be a very complicated system. Lots of ways to regulate it. Some are specific for cyclic A, some are specific for cyclic G, some hydrolyze both. Sometimes cyclic nucleotides regulate the hydrolysis of the other. An interesting story, an ideal place for drug targets. The phosphodiesterase that I'm sure you're all aware of and have heard about is the type 5 phosphodiesterase, which is found predominantly in blood vessels, which is selective for cyclic GMP. That's the target for Viagra. Viagra inhibits that phosphodiesterase, permitting cyclic GMP to accumulate in the vascular wall of the blood vessels in the corpus cavernosum. The blood vessels dilate, engorge with blood. That's the mechanism of penile erection. That's why it's used for erectile dysfunction. But there are other approaches. You can manipulate cyclic AMP levels or cyclic GMP synthesis. There are all sorts of ways to get there besides Viagra. But it's become very popular all over the world with almost $1 billion in sales. The cyclic nucleotides and most second messengers regulate various protein kinases. There are protein kinases that are selective for cyclic AMP, some selective for cyclic GMP, some are calcium calmodulin dependent. There are lots of these protein kinases. In fact, there are probably 150 protein kinases. Some phosphorylate serine threonine residues on proteins, some phosphorylate tyrosine residues. Most often, the tyrosine kinases are coupled to various cytokines and growth factors, such as platelet-derived growth factors. Some of them, in fact, are kinases. Most of the cyclic AMP and cyclic GMP kinases are serine threonine kinases that catalyze the transfer of the terminal gamma phosphate of ATP to a protein substrate. There are lots of protein substrates. When the protein is phosphorylated, its physical chemical properties can change if they're cytoskeletal proteins or contractile proteins, or if they happen to be enzymes, the activity can be increased or decreased, either way. So the concept of cell communication is rather straightforward and pretty simple, right? A ligand, a hormone, makes a messenger. The messenger activates the kinase that phosphorylates the protein, you get a physiologic response. The problem is that when you look at the matrix, it gets pretty hairy because there are many adenylate cyclases, there are many guanylate cyclases, there are lots of phosphodiestrases and lots of protein kinases and lots of protein substrates. And which proteins get phosphorylated depend on consensus sequences. It also depends on their compartmentation and their proximity to the protein kinase. Well, as this field got going in the late 60s, early 70s, I became very intrigued with it for several reasons. The cyclic GMP field, I thought, was becoming rather crowded. A lot of people, the technology was getting easier. I felt as a young faculty member going to Virginia, it would be difficult to compete with these factories, and I wanted to switch gears. And so I switched to cyclic GMP. And I wanted to figure out how hormones and ligands precisely regulated guanylate cyclase activity to make cyclic GMP. If we understood ligand receptor coupling to cyclase, perhaps we could come along with agents that would disrupt that coupling or potentiate that coupling to modify the message accumulation in various endocrinologic diseases. Could I come along with agents that treat Cushing's disease or diabetes or hyperparathyroidism or whatever? The second interest was to figure out what cyclic GMP is doing. Is it a messenger, truly? And if so, what does it regulate? And none of us had any clues. It was known at this point that acetylcholine in some heart preparations would increase cyclic GMP, and the prostaglandin would increase cyclic GMP in vascular preparations. But the agents failed to activate the enzyme in cell-free preparations. So that was a real challenge. If you're going to look at the mechanisms of coupling, it requires cell-free extracts that work, so you pull the components apart and reconstitute to really test the concept of their coupling. That was not doable at the time. We began our studies by looking at the soluble guanylate cyclase and the particulate guanylate cyclase in tissues, and we're quite surprised to find many tissues had both activities in the supernatant as well as the high-speed particulate. Unlike adenylate cyclase, all the activity was particulate, except for testis, where it's a fragment of the cyclase and some of it's soluble. We didn't expect to find activity both in the soup and the pellet. Furthermore, the kinetic properties of the enzyme and the soluble and particulate fractions are quite different. The greatest difference was the cooperativity of the particulate enzyme with regard to substrate. The double reciprocal plots were curvilinear, implying multiple interactive GTP sites. The soluble enzyme had typical Michaelis-Menten kinetics. We thought, my goodness, they're isoforms of guanylate cyclase, soluble and particulate. Wouldn't that be interesting if they had different regulatory mechanisms in different compartments of the cell to do different biological effects? This is going to be a fun project. Well, when you work with crude extracts, you can be misled by kinetic data because of a lot of potential artifacts. Our fractions contained nucleotidases, phosphatases, phosphodiestrases that competed for either substrate or product, so a lot of what we're seeing may have been bad data, fluky data, artifactual data. We knew that ultimately we'd have to purify these enzymes to homogeneity, clone them, express them, and re-examine them. And we've done that, but gosh, that took 10 or 15 years, and I wasn't patient at the time. So we took a shortcut. We rigged a cocktail of various goodies and threw into our incubations that would block these competing pathways. We made mixtures of pyrophosphate, fluoride, azide, hydroxylamine, nitrite, phosphodiesterase inhibitors, etc. And quite surprisingly, found that some of these substances activated the enzyme. Hormones would not activate, but we found that azide, hydroxylamine, and sodium nitrite would activate the soluble guanylate cyclase. The azide effect was oxygen dependent. It was tissue specific. It activated some preps, but not all. And it had a time lag before the enzyme activity became maximal, all of which suggested that it must be converted to something else in our incubations. So it became quite a detective story for a couple of years to figure out what that something else was. This is how we went about it. We did a very simple reconstitution experiment. We took a liver supernatant where azide would activate. We took a heart supernatant where azide had no effect, and a cerebral cortex supernatant where azide had no effect. And we mixed them because they were soluble fractions. We could get away with it. When we mixed liver with heart, the azide effect was blocked. That's because heart possessed an inhibitor. The inhibitor was heat labile, non-dialyzable, presumably a protein. We purified it. It turned out to be two proteins, hemoglobin and myoglobin. Mixed cerebral cortex with liver, the azide effect was enhanced. It was potentiated. And that's because liver converted azide to an activator, which would activate the cyclase contributed both by the liver as well as the cerebral cortex. The activator factor in liver, we purified, turned out to be catalase. So we now had a family of heme proteins that govern the tissue specificity of the azide activation. Why is that important? It'll become clear shortly. We took azide and hydroxylamine and nitrite and used that as a surrogate for hormone effects. And we dumped them on cell cultures and tissue slices. And sure enough, very often they would elevate cyclic GMP levels. So they worked both in cell-free preps as well as intact cell preps. Fortunately, as serendipity would have it, one of our preparations was tracheal smooth muscle. I wanted to correlate the biochemistry of second messengers with motility of smooth muscle, contraction, relaxation. And we developed a bovine tracheal smooth muscle prep that was relatively homogeneous, unlike vascular smooth muscle or other smooth muscle. So we can freeze clamp the tissue, look at enzymes, messengers, and correlate it with the motility state of the tissue. I thought that cyclic GMP would contract smooth muscle because I knew from my work here as a student that cyclic AMP relaxed. And I thought cyclic GMP would antagonize some of the effects of cyclic AMP. We put azide on the smooth muscle. It elevated cyclic GMP levels as expected. The big surprise was that it caused relaxation. That was unexpected. The relaxation had a wonderful time course of relaxation compared to the accumulation of cyclic GMP. The dose response curves fit quite well. So we were quite convinced over the course of a couple of months that one of the biological effects of cyclic GMP was to cause relaxation of smooth muscle. Not only airway smooth muscle, but gastrointestinal smooth muscle and subsequently vascular smooth muscle. We avoided vascular smooth muscle initially because it's so heterogeneous. You can't get a good, clean, smooth muscle prep to do biochemistry. Only about 30% of it, maybe at best, is smooth muscle. But once we figured out the pathway, the tools, we can now go to more and more complex models. When we found that these agents were relaxants, we said, my goodness, what happens with other smooth muscle relaxants? Well, the obvious ones to test were nitroglycerin and nitroprusside. Nitroglycerin was first synthesized in the 1840s by Italian chemists. It was first used clinically for angina pectoris in the 1870s. Alfred Nobel made his fortune by manufacturing nitroglycerin to make dynamite by mixing it with a diatomaceous earth so it was less explosive and safer to work with in his factories. Nevertheless, he still had some explosions and his brother died from one of those. But when he died, he left his fortune to create the prize, obviously. And he had angina pectoris and was prescribed nitroglycerin but refused to take it because his workers working with nitroglycerin would absorb it through their skin. They'd get vascular headaches, vasodilatation like migraine, and therefore he wouldn't take it. Well, he didn't die with a heart attack. He died with a big stroke. So probably just as well that he didn't take it. He might have had even a bigger stroke. He survived for about 1.5 years after his stroke. So we added nitroglycerin and nitroprusside to our systems, and sure enough, they caused relaxation, and they also increased cyclic GMP accumulation. So it now all fell together, and we developed this slide more than 20 years ago, showing you a list of agents that we called nitro-vasodilators. These are all prodrugs, or precursors, that are converted to what we thought was nitric oxide. They include nitrite, azide, hydroxylamine, nitroprusside, nitroglycerin, nitrosamines, nitrosoureas. There's a long, long list of NO donors. And they're all converted to NO in different ways, some spontaneously, some enzymatically. Why did we suspect they were being converted to NO? Because hemoglobin and myoglobin, the inhibitors we purified out of heart, would block their effects in all of our preparations. It was known from the literature that goes back to really the early 1900s that nitric oxide had a high affinity for heme. And we reviewed some of that was back in the 1940s and all. We said, my goodness, the intermediate's got to be nitric oxide. Well, we made nitric oxide in the fume hood chemically, ventilated the gas into our incubations, and sure enough, it activated every prep that we tested. And we were so excited because this was the first time that a free radical activated an enzyme. And we said, this has got to be exciting chemistry. Well, our colleagues didn't believe us. They thought this was nonsense, that we're destroying an inhibitor in our system. So we had to purify the enzyme to homogeneity to retest it. And when we did, we found that the concentration of nitric oxide required was even lower. As you remove proteins and transition metals and thiols as scavengers, the requirement for NO dropped to nanomolar concentrations. So very low concentrations of the free radical will activate guanylate cyclase, and the Vmax can be increased as much as 200 or 400-fold, a dramatic effect. And if we oxidize the NO to the dioxide or something else, it doesn't work. This is an example of what an NO donor, nitro-vasodilator, this happens to be nitroprusside, will do to a smooth muscle preparation. This happens via radiota segment, pre-contracted with norepinephrine, and then we add nitroprusside, and within 10 or 20 seconds, cyclic GMP accumulates. The accumulation precedes the physiology, that is the relaxation. The second messenger disappears before the physiology is gone. Why is that? Because downstream, the protein kinase and the phosphorylated proteins have a different half-life and turnover than does the messenger. It's a very common phenomenon. Messengers are often very short-lived, whether they be cyclic A or G or calcium or whatever, but then the downstream activities can persist for some 10 or 20 minutes or more. So we realized that we had figured out the mechanism of action of the nitro-vasodilators by producing nitric oxide to increase cyclic GMP. Well, as a pharmacologist, when you find an extraneous exogenous material that regulates some pathway or event, you should think that maybe it's mimicking some endogenous pathway. The opiates led to the discovery of enkephalins. There are many examples of that in the pharmacologic literature. And I said, is it possible, back in 1978, that hormones and drugs that elevate cyclic GMP are doing it by altering the production of nitric oxide from some endogenous precursor, perhaps by altering free radical redox chemistry and the conversion of some endogenous precursor to nitric oxide? Is nitric oxide an endogenous second messenger? Well, everybody thought I was really crazy when I proposed that. The problem is, at the time, we couldn't prove it. The reason we couldn't prove it is because the concentration of NO required was nanomolar concentrations, and we couldn't measure it at those concentrations. The assays for nitric oxide, nitrite, nitrate as oxidation products were very insensitive, 1,000-fold off the mark. So we had to spend the next 7 or 8 years developing new technology to prove to ourselves that we were right. Well, in the meantime, other laboratories became interested, and by the mid-1980s, there were all sorts of ways to measure nitric oxide or its metabolites in tissues. And it turned out this hypothesis was correct, and I guess one of the reasons I went to Stockholm. About this time, in 1980, Robert Furchtgott, a pharmacologist at SUNY Downstate, who was looking at blood vessels for a long, long time, vascular pharmacology, found for the first time that a handful of agents caused relaxation of vascular tissue in an organ bath. The agents were acetylcholine, histamine, bradykinin, a long list. Before, they had never relaxed smooth muscle in an organ bath, but we knew that they were all hypotensive when given to animals or man. It's because everybody was damaging the integrity of the endothelial lining of the blood vessels by handling the tissue when they were making their sections and mounting it on in the hooks in the organ baths. And if you were careful to preserve the endothelial integrity, the tissue relaxed. And Bob did a very classic, simple experiment. He took a blood vessel with endothelium and sandwiched it up against a blood vessel without. And when he added acetylcholine, both strips relaxed. So there was an endothelial factor that caused relaxation of smooth muscle. It had a short half-life of several seconds. He presented this at a seminar at Virginia. He also found that sunlight, would cause relaxation. We had been playing games with photochemical activation of Guanylate Cyclase in some of our laboratory experiments. And I said to Bob, this has to be through cyclic GMP, and it's probably a free radical. Could be nitric oxide, but maybe another free radical. Let's collaborate. And we agreed to collaborate. Well, he went back to New York. His wife developed breast cancer. We did not collaborate. We moved to Stanford. And after about a year, one of my fellows became very impatient waiting for samples that we never got. And we went ahead and did it ourselves, and sure enough, we were right. An endothelial-dependent vasodilator, such as acetylcholine, bradykinin, thrombin, histamine, all sorts of things, ionophore, will only cause relaxation if the endothelium is intact. And when it is, it also elevates cyclic GMP levels in the smooth muscle compartment. If you take off the endothelium, there's no increase in cyclic GMP, no relaxation. Well, Furchtgott called this substance EDRF, endothelial-derived relaxin factor. He and I have argued for years as to what it might be. I think it's a mixture of things. He and Ignero and Moncada think it's NO. I think NO is only one of many components of EDRF. It's probably a lot of other things as well. But at such low concentrations, I doubt that we'll ever resolve it and figure it out. So by the early 1980s, we knew that we could activate cyclase, guanylate cyclase, through NO, as well as through the production of EDRF, which we now know to be really converging pathways and are probably the same. So we spent a couple of years looking at downstream effects. We measured endogenous cyclic GMP-dependent protein kinase activity in vascular preparations and cell cultures as well. We even looked at P32 incorporation of proteins and displayed them on 2D gels, and we were able to identify some of these proteins and put this story together for you. This cartoon represents a blood vessel with an endothelial lining and an underlying smooth muscle compartment. In red are 3 categories of vasodilators that function by increasing cyclic GMP production. The nitrovasodilators spontaneously release NO based on the local pH, oxygen tension, whatever in the bloodstream, or they're enzymatically converted to NO, as with nitroglycerin. It requires an enzyme to denitrate it. The NO binds to a heme prosthetic group on the beta subunit of soluble guanylate cyclase, which is a heterodimer. That changes the conformation of the protein, and at the C-terminal, the catalytic domain, which is redundant between the alpha and beta subunits, participate in the catalysis to make cyclic GMP. The cyclic GMP activates cyclic GMP-dependent protein kinases, and there are a family of these. Some are soluble, some are particulate. They phosphorylate a variety of proteins that you can chase on 2D gels with proteomic methods, which we did. Some of these proteins regulate cytosolic calcium by decreasing calcium, which in turn decreases the activity of myosin light chain kinase, which is a calcium calmodulin-dependent enzyme, resulting in the dephosphorylation of myosin light chain. When you dephosphorylate myosin light chain, the actin-myosin filaments slide apart. When you phosphorylate the myosin light chain, They slide back and you get contraction. The endothelial-dependent vasodilators do exactly the same thing. But the receptors for the endothelial-dependent vasodilators are only on the endothelium. They're not on the smooth muscle. So therefore, the nitro vasodilators are direct acting. The endothelial-dependent vasodilators require the integrity of the endothelium. There are diseases where the integrity of the endothelium is abnormal, and I'll tell you about some of those. That's why we use nitro vasodilators in the intensive care unit in patients with heart attacks or atherosclerosis. They'll work independently of the endothelium. They enhance calcium uptake in the endothelial cell, which activates a family of enzymes called nitric oxide synthases that convert arginine to EDRF, or nitric oxide, which then moves over to the smooth muscle compartment to have the same pharmacology and biochemistry. The third category are the atropeptins. When Pilati was looking at the organization of cells histologically, he found that cardiac atria possess granules. When you see granules in cells, that's often indicative of stored peptide hormone, chromaffin tissues, other sorts of tissues. This attracted the attention of DeBold in Canada, who isolated these granules, purified the peptide, sequenced it, and called it atrial natriuretic factor, or it's now called atropeptins, and they're a family of these peptides, not only from heart but other tissues as well. We saw this work, and we became quite intrigued with it because the atropeptins were functioning as vasodilators in animals to lower blood pressure. They were also natriuretic to enhance electrolytes excretion by the kidney. Some years earlier, we had been looking at heat-stable enterotoxin, STA, and its mechanism to cause diarrhea when I was at Virginia. And we found that that heat-stable enterotoxin activated a particulate isoform of guanylate cyclase, GCC, found only in the intestinal mucosa and some species in the kidney. It altered ion transport. By making cyclic G, you activated the kinase, you phosphorylated CFTR, chloride came out, as did sodium water, and that was the mechanism of diarrhea. Well, today we found a drug to block that pathway. I'm very excited about it. It's a therapeutic, perhaps, for diarrhea in third world countries, and we're pushing pretty hard on it. But when the atropeptins came along, they had some of the features of the enterotoxin on ion transport, but they had some of the features of the nitrovasodilators on smooth muscle relaxation. And we said, we better check these on cyclic G, and sure enough, they activated the particulate Guanylate Cyclase, but they activated a different isoform, GCA. Well, the atropeptins, by activating the particulate cyclase, it turns out that all of the particulate cyclases are receptor cyclases. They're transmembrane proteins with receptor domains on the outside, a short membrane span, catalytic domains on the inside. They make cyclic G and also result in relaxation of smooth muscle. Well, about this time, Deguchi, the director of the Neuroscience Institute in Tokyo, was also interested in guanylate cyclase in neuroblastoma in brain and was purifying his preparation and found that the specific activity of his preparation didn't quite match the intensity of his Kumasi spots on gels. It's because he had an activator in his prep. The activator he identified is arginine. Arginine will activate crude and partially purified preparations of soluble guanylate cyclase. It does not activate purified guanylate cyclase. Arginine was somehow being converted to an activator, an important tip-off. John Hibbs, an infectious disease person at Salt Lake, was interested in the cytotoxic properties of macrophages in co-cultures with tumor cells. It was known that if you added pro-inflammatory cytokines and endotoxin to macrophages, you enhance their cytotoxicity. Well, you could also add arginine and enhance their cytotoxicity, and when you did that, you accumulated nitrite and nitrate in the conditioned media. And if you added arginine analogs, as he did, methylarginine, you block the cytotoxic properties of the macrophages. So it now became apparent that there was a pathway that entailed arginine to make nitrite and nitrate in systems, and that arginine analogs block that pathway. Therefore, there must be a pathway metabolizing arginine to some NO-like product. And we all went running after this new pathway. And it was a furious time with about 6 or 7 labs all hustling to get there first, in a very challenging time. And sure enough, there are a family of enzymes called nitric oxide synthases that convert arginine to nitric oxide. They were initially called neuronal NOS, inducible NOS, and endothelial NOS. I suggested we call them NOS 1, 2, and 3 because these enzymes are very ubiquitous and not just in the brain or the endothelium. They're all over the place. And they're numbered in the chronological sequence in which they were first characterized, purified, and cloned. There are 3 different genes that encode for these isoforms. There's a lot of homology, 50 or 60% between them. They're widely distributed. NOS2 is usually not present in tissues unless there's been an inflammatory insult. The gene is silent until it sees a pro-inflammatory cytokine, and then it's expressed to make nitric oxide synthase, and it makes a lot of NO. So it's become a marker of inflammation, if you will, and maybe a participant in the process. Why does it do that? Well, it may be doing that as a defense mechanism to kill pathogens, has been suggested. This is the reaction that all of the NOSes catalyze. They oxidize the guanidino nitrogen of arginine to an intermediate hydroxyarginine, which is further oxidized to NO, and the other product is citrulline. The enzymes have a very complicated array of cofactors. They have a heme prosthetic group. They're only active as homodimers. They utilize NADPH, oxygen, flavines, tetrahydrobiopterin, calcium calmodulin. Why all the complexity? I think there's a reason for that, and to control their regulation. So you can control the activity of these enzymes by controlling all the cofactors or the expression of the protein. This is really, which allows you a lot of approaches in terms of thinking about drug development to inhibit or activate these pathways. Let me put this together for you now. In a very simple version, various hormones and ligands will interact with their appropriate receptor, and very often in this pathway they alter the availability of a cofactor, very often calcium calmodulin, but perhaps tetrahydrobiopterin as well, that permits these enzymes, nitric oxide synthases, to make nitric oxide from arginine. The NO activates the soluble cyclase, which can go on to activate a kinase to phosphorylate proteins to get a physiologic effect. You can interrupt this pathway or mimic it in a variety of ways. You've got receptor agonists and antagonists. You can manipulate the cofactors. You've got analogs of the substrate that are competitive inhibitors. You have scavengers of NO, some simple molecules, some large complicated molecules. There are various ways to influence the activity of guanylate cyclase, either basal activities or the activatability by NO. And you can certainly influence the kinases and the phosphodiesterases. So you've got a lot of tools now to go testing biological models, animal models, and to think of them as possible therapeutic targets to discover novel compounds. There's more to this pathway. There are other fates of nitric oxide. Nitric oxide, I think, is primarily made to activate guanylate cyclase to make cyclic GMP to do good things, to inhibit platelet aggregation, to dilate blood vessels, enhance blood flow, to function as a neurotransmitter, et cetera. But it has toxicity too. It's converted to nitrites and nitrates, which have no activity, but they can be recycled back to make NO. The salivary gland entraps nitrate. Your saliva converts it to nitrite with a bacteria in your mouth. You swallow the saliva, it goes to your stomach, the nitrite goes to NO in the acidic stomach. The salivary glands are talking to the stomach. Pavlov missed that one, right? It also can form complexes with other transition metals, not just iron, which it prefers, but other transition metals, manganese and others as well. And it can form nitrisation products with cysteine thiols. There are about 150 proteins that get nitrosated on their thiol groups. Some are very important, the NMDA receptor, some of the transcription factors, some of the caspases, lots of other things as well. Now, a very important reaction is the interaction of nitric oxide with the superoxide anion. You accumulate superoxide anion most often with inflammatory insults as you influence NADPH oxidase, xanthine oxidase, prostaglandin metabolism. The superoxide anion has a very high avidity or affinity for nitric oxide. In fact, the interaction is almost diffusion limited. If you make NO in the vicinity of superoxide, it's all going to go to peroxynitrite. It won't have an opportunity to activate G cyclase. What is the reason for this? Well, I don't know. But we do know that peroxynitrite is very toxic. And one of the readouts of peroxynitrite formation is the nitration of tyrosyl residues in proteins. And we've identified probably 20 or 30 proteins that get tyrosine nitrated adjacent to the hydroxyl group. And we wonder if that nitration is changing the pK of the hydroxyl and therefore modifying their ability to be phosphorylated, or is it sterically blocking phosphorylation? Does the NO pathway talk to tyrosine kinase? I think it might, but it's been a bear to prove. And the only way to prove it is in intact cell or in vivo systems. You can't just do it in extracts in the laboratory. And we do occasionally see it, and we're finding some now with regard to transcriptional regulation that are getting us quite excited. But it's early, don't know. Most people have used protein nitration as a footprint or readout or biomarker of inflammation. When you see it, there's inflammation. If there's no inflammation, you still see it, but it's not as prominent. And you can follow it in various ways. You can do this with the cleavage of the protein in mass spectroscopy. You can do it with antibodies using the nitrotyrosine as an epitope, and all sorts of ways to do it. Now let me try to relate this to some real clinical medicine for you. I don't know how much more time I have. I need to ask here. Am I almost through here? I'll hustle. There are a number of diseases, hypertension, atherosclerosis, diabetes, tobacco use, probably obesity, that have what is called endothelial dysfunction. Their blood vessels don't make enough nitric oxide. Why is that? All of these diseases are associated with reactive oxygen species production. You have oxidative stress. You make free radicals, hydrogen peroxides, superoxide, maybe hydroxyl radical. All of these oxidative species are going to screw up the cofactors required for nitric oxide synthase. If you oxidize the tetrahydrobiopterin to dihydro, the enzyme no longer makes NO. Instead, it makes superoxide, which makes the situation worse, right? So, and the other thing that happens in all of these diseases is that you make an inhibitor of the pathway, asymmetric dimethylarginine. One of the pathways to process proteins is the methylation of the arginine guanidino nitrogens. And if you put two methyls on the same nitrogen, you form asymmetric dimethylarginine, which is a competitive inhibitor of the catalytic site of nitric oxide synthase. So you've got to get rid of the asymmetric dimethylarginine, and in all of these diseases, it's elevated. So there's some rationale now, I think, evolving to explain perhaps the benefit of arginine supplementation, the benefit of antioxidant therapy. You know, some of the traditional Chinese medicines, which I've become interested in, maybe make some sense. I don't know. We've got to do a lot more. There's a lot of laboratory data, animal data, to support all this. There's very little clinical data. And what clinical data is there is sort of controversial. Some's positive, some is negative. So it's still to be resolved. Let me go through a list of all sorts of diseases where this field has led to the development of various drugs that are most of which are in clinical trials. Some are still at the bench, and a couple are already on the market. And I'll go through them quickly. And I've written a couple of reviews. The most recent was the New England Journal Shattuck lecture this past year. You can go there if you like to see this. I'm not going to talk about it all, let me pick a couple. Pulmonary hypertension. In premature babies, they maintain their fetal circulation. In their fetal circulation, they don't need to perfuse their lungs because they get oxygenated blood in the placenta from the mother. So they shunt blood right to left. They perfuse the lung, but not very well. The blood goes through a patent foramen, the patent ductus, into the systemic circuit. They're hypoxic, blue babies. How do you treat them? Well, you used to treat them with extracorporeal membrane oxygenation, ECMO, rather a devastating, horrible procedure. A lot of strokes, a lot of problems. However, if you give them very low concentrations of inhaled nitric oxide, you dilate their pulmonary vessels. They now perfuse their lungs. They don't have the perfusion aeration mismatch. The blood gets oxygenated. They're less hypoxic. They do fine. And after a short period of days or even a few weeks, along with surfactant and other support, they seem to do pretty well. It's really been a marvelous therapy. We talked about penile erection. We know it plays a role in angiogenesis and wound healing. We've been doing a lot of studies with that. We know that we think it's going to be beneficial in diabetic foot ulcers with topical applications. We know that there's inflammation in atherosclerotic plaques. You can see a lot of nitrotyrosine in those plaques. It's probably because you're inducing NOS2, a lot of other reactive oxygen species. In asthmatics, you can measure nitric oxide exhaled in the breath, and the more exhaled, the worse the asthma. It's a good predictor for the management of asthmatic patients. It's becoming very popular, and that's because asthmatic patients get bad when they have inflammation, infection, and what they do is they induce NOS2, make a lot of NO, and they exhale it. Well, you got to be sure that it's coming from the lungs and not from the nasopharynx because you have a lot of NO in your nasopharynx to keep them clean. It's very rich in nitric oxide synthase. Another trick, platelet aggregation. There is a young cardiovascular surgeon in Germany who told me a couple years ago that he's infusing nitric oxide gas in the cardiac bypass machine. Why does he do that? Well, the bypass machine damages the platelets, they aggregate, and the yield of platelets coming through the machine diminishes with the aggregation. By spritzing in a little NO, he prevents the aggregation. More platelets come out, more go back into the patient. His OR time is diminished because there's less bleeding into the chest. As the patients recover faster. Simple technique that has really influenced patient care. Not approved yet, but it should be. Stem cells. We're playing a lot with stem cells. We know that stem cells express all the components that we've been talking about during the early phase in embryonics with mouse or human, through maturation into cardiomyocytes, neuronal cells, glial cells. We've got the whole profile of NOSes and cyclases. By having that profile, we can now come in with cocktails of pharmacologic agents, manipulate those cells, and make them go into certain lineages. And that's going to be fun. How can we make them all become neuronal cells or all myocardial cells in terms of future transplantation? I'm going to stop. I've gone over. I'm sorry. Do we have time for a couple questions? Take it all.
Who's in charge here? Do we have time for questions?
Yes. So, yeah. I had two questions. One is, can ammonia be converted to nitric oxide? And the other issue is that there's a rare genetic disease, arginase deficiency, and the arginine levels are extremely high. And they have a very unusual phenotype for urea cycle defects. So I wondered if any of this is due to.
What was the first question?
Can ammonia be converted to nitric oxide?
I don't know that humans or mammals can do it. I would guess that bugs could, but I don't know. I don't have the answer. And with regard to the arginase deficiency, I don't know much about the phenotype of that disease.
It's like a neurological disease.
Okay.
Movement and gait disorder.
I guess I can't answer either